The Experimental Toxicology and Ecotoxicology Unit is a technological platform of the Barcelona Science Park. And as such, as a technological platform, we are in the frame of an institution that allows us to gain access to infrastructure, scientific infrastructures and technologies that normally would not be accessible for a small lab as ours. So we may gain access to electron microscopy, confocal microscopy, or atomic forces microscopy, or flux cytofluorometry, etc. That greatly improves our possibilities to perform toxicological assessment. Our offer is focused on the design and building of batteries of tests, basically short-term in vitro tests, adapted to the needs of our clients and able to cover the different blocks of relevant information in toxicology. It is systemic toxicology, toxicology of the reproductive process, genotoxicology, or cancerogenesis. To design such batteries, we have implemented in our lab quite a wide array of techniques, as for example to cover the systemic toxicology information, the studies of cytotoxicity with different cell strains and with different endpoints, neutral red uptake or MTT or WST. For genotoxicity, we focus mainly, but not only, in two techniques, the micronucleus assay and the COMET assay, or single-cell gel electrophoresis. That, in my advice, is a very interesting and very versatile alternative to micronucleus. In the field of reprotoxicity, of the embryo toxicity, we have implemented the three techniques that are presently validated at the scientific level by the European Committee for Validation of Alternative Methods. I mean the embryonic stem cell test, rodent embryonic stem cell test, DST, that is a technique based on the potential, the capacity of the the product test to interfere with the differentiation of pluripotential cells to become differentiated myocardiocytes, cells of the heart, and so to be able to contract, to be contractile cells. The micromass test based on the primary culture of chondrocytes, embryonic rodent chondrocytes, and which endpoint is to test the capacity of the toxic to interfere with the differentiation of such embryonic cells to cartilage nodes and the whole embryo culture. I mean the culture of whole rat embryos in vitro medium during 48 hours, during which we expose them to the test compounds and we assess the capacity of such compounds to produce malformation. For cancerogenesis, we mainly use, not only, but we mainly use the cell transformation assay. That is an assay based on the capacity of the products to phenotypically transform normal cells to cancerogen cells in two steps, initiation and promotion. So this test enables us to test the compound in both aspects, as an inductor, as a product able to initiate the carcinogenic transformation, or as a promoter of mitogenic compound. For ecotoxicological assessment, we have implemented the OECD protocols for terrestrial medium compartment and for aquatic compartment. And we perform them under good laboratory practices. For the terrestrial compartment, we test the capacity of nitrogen fixation by the bacteria of the soil, the germination and growth terrestrial plant test, and the fertility and reproduction test in earthworms. For the aquatic compartment, we test the compounds in different strains of algae, of green or blue algae, independent of the different kinds of compounds to be tested. Invertebrates, aquatic invertebrates, such as Daphnia magna or Nitocra for salt water, for marine water, and also in fish, in zebrafish or in marine species. As all the other platforms in the Barcelona Science Park, a very important part of our activity, on the other hand, is basic research. So we perform basic research with public funding and in competitive projects, both at the national and at the European level. We are currently involved in 8 different competitive projects that cover quite a wide array of different topics, going from the assessment of toxicology of nanoparticles or nanomaterials to the modeling of how to introduce the eco-surveillance data in the integrated risk assessment for humans from environmental stressors, or also a project, for example, on the sustainable cycle of water.